So now what we're going to do is we're going to put ourselves into the simplest possible setting. And our mathematical model is going to be based on that setting. So this may seem a bit strange, but bear with me. I'm going to make a series of assumptions. It's imperative that you realize that these assumptions are not things that I'm saying are true. They're not things that I'm saying I think are true. They are things that I'm saying suppose these were true for the sake of argument. Now, if you allow me that argument, then I can predict what evolution is going to do. I can predict, is P going to increase, decrease, or stay the same? Now, it is very restrictive, but that's the point. It becomes very restrictive. So bear with me, because I'm going to make a very important point, even though the assumptions I'm going to make are going to be so restrictive that it's going to limit its utility. It actually doesn't. It makes it really, really broad. So let me explain. Suppose in our model, we're considering a population with the following properties. There's 5 of them. First, I'm going to assume that the population is large. Now, technically, I really should be assuming the population is infinite the way I'm presenting it. Your book says that. But really, actually, we can show with a little more sophisticated mathematics that it doesn't have to be large. It just has to be, so it doesn't have to be infinite. It just has to be large enough so that random fluctuations associated with small population sizes are negligible. All right, so we're going to make that assumption. Population is big. How big is big? In reality, it depends on a lot of things that are going on, but if we're looking at populations in the sizes of millions, that's big. That's certainly big enough. The second assumption we're going to make is that mating is random. And now this is a little bit confusing. Let me be clear. We're not saying that people just mate with whomever. People do choose their mates, and they choose their mates based on traits that they like. But what I'm saying is they're not choosing their mates based on the trait at hand. Now we're going to be talking about TPA25, whether or not you have the TPA25 on both chromosomes, on one chromosome only, or on neither. Right? So let me ask you this. When was the last time anybody walked up to somebody else in the bar and said, "Hey baby, what's your TPA25 status?" I mean, that doesn't happen. So that's the kind of thing we're talking about. It's the trait that is not involved in who's mating with whom. Okay, third assumption is the population is closed, which means no one is emigrating from the population or immigrating to it. Okay, so there's no gene flow, there's no migration that way. And next assumption is that mutations in this gene are so rare that we can ignore them in the long term. Now, the key phrase is in this gene. We're not saying that mutations don't happen at all. We're not saying mutations are not something that occur elsewhere in the genes. We're saying that this particular gene doesn't disappear. The TPA25 doesn't disappear randomly very often, and it doesn't appear randomly in the exact spot that we're studying very often. Okay? And in fact, that's a fairly reasonable assumption. And then finally, the last one is that there's no natural selection, meaning having this TPA25 is not beneficial, it's not detrimental, it's totally neutral. Okay, now, you might be saying, and you should be saying, wait a minute, I thought that's what we were trying to test. You're right, we're trying to test it, but remember, what I'm saying is assume that these statements are true. Assume that these assumptions hold for the sake of argument. Okay? Now, if you think about it, I can ask this. In what populations do all 5 of these assumptions hold? Does that happen very often? Now, if you think about it carefully, even in this population, we're talking about northern Italy, right? In northern Italy, is the population large enough that random fluctuations are negligible? Well, yeah, there's millions of people there, so yeah, sure. What about mating is random? Well, again, like we established, people don't ask each other what their TPA25 status is. On the other hand, we don't know. The TPA25 might be associated with something that's attractive to people. We don't know yet. So we don't know about that one. What about no one entering or leaving the population? Yeah, that's the one. People are coming into northern Italy, they're leaving northern Italy, it's not a closed population, so this one definitely fails. What about mutations are so rare that they can be ignored? In this particular case, I don't expect you to know the answer to that, but in this particular case, it's extremely unlikely that an ALU insertion would put itself into that exact range that we're studying by chance. It's even less likely that it would just remove itself by chance completely. So that one's probably okay. Natural selection, that's one we don't know about. Okay, so if these 5 assumptions hold, though, for the sake of argument, I can tell you what evolution was going to do if these 5 assumptions hold. And here's how. It's relatively straightforward, but I'm going to do something. I'm going to do something that scientists do that the general population somehow thinks is cheating. It somehow thinks it's not fair or it's not right, it's flawed logic, and it's not. It's absolutely perfect logic. What I'm going to do is I'm going to take this crazy, chaotic situation of people in northern Italy breeding together over generations, and I'm going to look at it in a different way, which is logically identical, but gives us a much easier solution than trying to work through all of the chaos. Think about it. If in order to predict evolution, what we got to do is figure out who's going to mate with whom, with what probability, because every single individual could have a multiple number of people that they could mate with, and we have to figure out the probabilities that they mate with those people. We have to then figure out the probabilities of their offspring, put all of that together. That's insanely complex. But we're not going to do that. We're smarter than that. What we're going to do is we're going to look at the exact same problem, but from a different perspective, which is a fiction. But the logic is identical. Okay? Now, this is one of those things that you just have to see. And I'm sorry, I can't teach you to see it. I don't know how. You see it or you don't. And this is one of the things that I can prove. I can prove that everything I'm saying is correct. But to do that requires mathematics, which is a bit beyond where we are. It's not wildly complex mathematics or anything, but it's beyond where most of us are in our mathematical studies. So the proof would be useless. It'd be just gibberish. So I can prove it, but proving it doesn't help. But the point is you can see it. You can see that what I'm about to say is logically identical to all this chaotic breeding. Okay? Take those 5 assumptions. If it's true that these 5 assumptions hold, then all of those matings that we would have to try to predict come down to this. Logically, what I can do is take all the women in northern Italy, line them up, take the first one in line, do a genetic test on her, and determine whether she's plus plus, plus minus, or minus minus. If she's plus plus, what I can do is take 2 red poker chips, put a plus symbol on them, and throw them into a barrel. If she's plus minus, I take a red poker chip with a plus and a blue poker chip with a minus, throw those in the barrel. If she's minus minus, you see, I'll bet you see it, 2 blue poker chips, both with minus, throw those in the barrel. Okay, next woman, same thing. Put her poker chips in the barrel. Next woman, same thing. Do that for everybody. And in fact, I don't even really have to do it for everybody because I know already what proportion is already in the barrel. What proportion of red plus poker chips are in the barrel? Well, we just calculated it. We estimated it from that sample. The proportion that are in the barrel is that 0.4848 of plus that we saw. Okay, so that then, 0.5152, would be the proportion that are minus. So if I were to do this in Italy, that's what I'm going to get. I already know that from the sample. That's my estimate. Now, if I did it for the men, same exact thing. The men come down in a line, I test them, throw their poker chips in the barrel. When I'm done, the barrel is going to contain 48.40% of the chips are going to be plus plus, or just plus, and 51.52 are going to be minus. Okay, so that's what we're doing. Okay, so I already know, based on just that logic, that all of those chaotic breedings come down to simply this. A mating is randomly choosing one poker chip because they're not choosing each other based on the poker chip. They're not choosing each other based on whether they're plus or minus. There is a huge population, so every possible combination is going to occur at its normal frequency. So all of that just boils down to having a baby in that population means taking randomly a poker chip from the female barrel, which represents an egg, and taking a poker chip from the male barrel, which represents a sperm, putting them together, and that's the kid. Now, that's logically the same. It comes out to be exactly the same if all those 5 assumptions hold in northern Italy. There's no other way to say it. Now, I can prove it. Like I said, I can prove it. But again, the mathematics are a little bit beyond where we are. But if you want to see the proof, it's in many books, including one that I wrote with researchers down at ASU. So the point is, it's there. Now, if you can't see it, trust it for now, but don't entirely trust it. Don't ever trust anything. Work through the proof. Learn how to do the proof and show yourself that that's the way it ends up being. But the point is, this is easy now. It's simple because all we have to do is this. What's the probability that I pull out a positive egg, an egg with a plus on it? Well, you already know it's 0.4848. What's the probability that I take a sperm cell with a plus on it? That's also 0.4848. So what's the probability that I get an egg that's plus and a sperm that's plus? Remember what and means mathematically. Mathematically, and logically means times. Okay, are they independent? Yes, because the egg that I choose is independent of the sperm that I choose, more or less. Right? So I can answer this question. What's the probability of TPA plus sperm? That's 0.4848. What's the probability that it fertilizes a TPA plus egg? 0.4848. So therefore, the probability is simply the product 0.4848 squared. Okay, that's it. Which means also this, we can do the entire problem using a Punnett square. It's really straightforward. In this case, you've seen it before, but what we're going to do is expand a binomial. That's what a Punnett square is. It's that FOIL. It's just a more complex, kind of convoluted way to see it, doing it, but it's easier to see what's happening. It's just clumsy. All right, so what we're saying is this. We know that in the egg barrel, okay, so that's the first gamete, that's the egg barrel, then it has two different kinds of poker chips, plus and minus. Okay, so there are the two different types of poker chips. I put those up there. Now, on this side, I'm going to put the barrel representing the sperm, which also contain TPA plus and minus poker chips, red and blue poker chips. Okay, now what this represents, this box represents a TPA plus fertilizing a TPA plus egg, right? So which means the offspring would be plus plus. In this case, the sperm carrying the plus fertilizes an egg carrying a minus, so that makes it TPA plus minus. This is also going to be TPA plus minus, but in this case, the egg brought the minus and the sperm brought the egg brought the plus. Pardon me, sperm brought the minus, and in this case, a minus sperm fertilizes a minus egg, and you get TPA minus minus. Now you've seen this before, but it's dangerous. This is not the same thing that we were doing before. What we were doing before, we were looking at individuals. This represented one individual, and this represented another individual. Here, this is representing everybody, all their possible gametes, and this is everybody also all their possible gametes. So in this case then, we have to keep track of the proportions because it's not equally likely that you end up with plus and a minus. To get a plus, what's the probability that it came out as a plus? The egg came out plus as a plus. Well, you know that's 0.4848. What's the probability it's minus? 0.5152. Okay, same thing for the sperm. What's the probability it's plus? 0.4848 minus is 0.5152. Great. So now what's the probability then that I get a plus sperm fertilizing a plus egg? Well, it's 0.4848 squared, which if you crank that out in your calculator, which you should, you get 0.2350. Okay? Now, 23.5%. Don't let that go by. Don't move on until you know exactly what that means. 23.5% of what are what if what? Okay? Stop the video. Think that through. Write down the answer. 23.5% of what are what if what? Okay, if you have the answer, here's what it is. This says 23.5% of the people in the next generation should be plus plus if those 5 assumptions hold. That's what that says. 23.5% of the people in the next generation should be plus plus if those 5 assumptions hold. Now, what if the 5 assumptions don't hold? Well, then all of the argument that we made about the barrels and all that stuff falls apart. So that means we probably get something different. That's critical. All right, so let's continue on though. So now what's the probability that I get a heterozygote when the plus is carried by the sperm? Well, it's going to be 0.4848 times 0.5152, which is 0.2498. Now, what about this though? Well, this is going to have to be the same numbers, 0.2498, because it's still 0.4848 times 0.5152. The only difference is in this case, this egg is carrying the plus. And this one is going to be 0.5152 squared, which is 0.2654. Okay, so all those numbers mean what? Well, here's what they mean. Here's what we expect. In the next generation, about 23.5% of the individuals, the people in the next generation, should be TPA plus plus if those 5 assumptions hold. Now that's going to be always the case here because we're just making the assumptions that the 5 assumptions hold. Okay, but now what proportion I should say, what percentage should be heterozygous? Okay, well, when we look at the heterozygotes, you can be heterozygous in 2 ways, either this way or this way, where the plus came from the sperm or the plus came from the egg. So that means this. What's the probability of heterozygous? Well, I could be heterozygous this way or this way. Remember, or means plus. Therefore, I take 0.2498 plus 0.2498, which is simply 2 times 0.2498, and I get 49.96% of the next generation should be heterozygous, if those 5 assumptions hold. And finally, the remaining 26.54% of the next generation should be TPA minus minus if the 5 assumptions hold. Okay, so that's going to be our solution. Now we've made the prediction based on the data that we got from Silvaggi et al. We've made the prediction of what we're going to see in the next generation based on this mathematical model. Now, we can do this more generally. Suppose we have any gamete, I don't care what it is, it doesn't have to be TPA, it could be something else, and I'm just going to call it A. And so it's got 2 alleles, big A and little a, another one big A and little a, which means we've done the Punnett square before. This is big A, little a, blah, blah, blah. Now, in this case, I'm going to be general. I'm not going to say it's 0.4848, I'm going to say it's P. It could be any number between 0 and 1, and it's P. Okay? And if you remember, we said this was Q. So the proportion of the barrels in the alleles in the barrel are going to be A is P. The proportion that are little a is going to be Q. And the same thing for the other barrel. Okay? So what is this then? Well, the probability of getting a homozygous big A is going to be P squared. The probability of being big A, where the little a is carried by the egg, is going to be PQ. The probability of being big A, where the big A is carried by the egg, is going to be also PQ. And the probability then of being little a is going to be Q squared. Okay, now that leads us to this statement. If the population is large, mating is random, there's no migration, negligible mutation, and no natural selection. Where did we see those before? Okay, those are the 5 assumptions that we saw before, the very first assumptions that we started with. If those things are true, then in the next generation we expect the following genotype frequencies based on the allele frequencies. In the next generation we should see the frequency of big A should be P squared. The frequency of big A should be 2 times P times Q. Remember that? Because there's 2 of these boxes. And finally, the frequency of little a should be Q squared. So what you're looking at here is what we call the Hardy-Weinberg theorem for 2 alleles at 1 locus. Here's a summary of what we've done so far. Remember, we started with 263 individuals in that sample that Silvaggi took. And in that sample of 263 people, 56 were plus plus, 143 were plus minus, and 64 were minus minus. Okay, I want to put that into terms of proportions because that's what seems to be changing. So let's look at that. What proportion of that original sample was plus plus? How would you get that? You know, at this point in your career, you should know how to do it. To get the proportion who are plus plus, it's straightforward. Take the 56 individuals who are plus plus, divide by the total 263, and if you do that, you get this. Go ahead and do that on your calculator, and you'll get this number, 0.2129. All right, so this says 21.29% of what are what? Always answer that in your head before you move on. 21.29% of the people in the original sample were plus plus. Okay, now, how many of them were heterozygous? Well, again, you take the 143 heterozygotes, divide by the total 263, and you get 0.5437. So 54% of the original people in the sample were heterozygous. Okay, and then to get the proportion who are homozygous minus minus, you take the number who are minus minus, which is 64, divide by the 263 individuals, and you get 0.2433. Okay, 24.33% of the people in the original sample were minus minus. All right, those are what we call the genotype frequencies. What are the allele frequencies? Okay, now remember, to get the allele frequencies, we took the total number of individuals who were plus plus, multiplied by 2 because each is carrying 2 pluses, and then took the number who were heterozygous and multiplied by 1 because they're only carrying 1 plus, and then divided that sum by the entire population size, 263 times 2, because everybody in the population is carrying 2 alleles. Okay, we've done that before. Okay, if what I just said is confusing, you're like, I'm not sure where they came from, stop, go back to the beginning of this lecture and go through that again. Okay, from that, remember, we found that 48.48% of the alleles, not the people, but the alleles in the population are plus. So the P, the proportion of plus, is 0.4848. And then the Q, the proportion that are minus, is 0.5152 because it's 1 minus 0.4848. All right, now my question is this. Did this population evolve? Well, what we just calculated was the frequency of individuals who are plus plus should be p squared, this number squared, which we did back up a little bit ways. And so we got 23.5%. Remember, that's where I was asking 23.5% of what or what if what. And the answer was 23.5% of the next generation, generation 1 from generation 0, should be plus plus if the 5 assumptions hold. And what is the expected frequency for plus minus? That's going to be 0.4996. We calculated that. Go back and see that if you're not sure where this is coming from. And then the same thing with minus minus. The frequency of that should be 0.2654 if the 5 assumptions hold. Okay, so did the population evolve? Well, look at this. In one generation, the proportion of individuals who were plus plus went up. Went from 21+% to 23.5%. The proportion of heterozygous went down, 54.33% about, almost to 50%. And then this went up, 24, a little less than 1/4% to a little bit more than 1/4 25%, 26%. So that appears to have changed, and there's no question it did. This is predicting it'll change. But is that evolution? That's the question. So did this population evolve? Okay, well, remember, what's the definition of evolution? Evolution is defined to be a change in allele frequencies. These are genotype frequencies. These are the allele frequencies. Did the allele frequencies change? That's what we need to know, and we don't know that. Right? So we haven't calculated the allele frequencies for generation 1. Okay, so let's do that. Let's go ahead and do that. In generation 1, here's what we know. This number here, 26.54%, 0.2654, we got by taking q squared. And we got that because of the Hardy-Weinberg theorem. Frequency of minus minus in this case is going to be q squared. So minus minus was q squared. Okay, so there is just simply 0.5152 squared. All right, so if that's true, how do I find q? If I know that the frequency of minus minus in this case is q squared, give me q. What's q? Well, straightforward. You guys know how to do this. Take the square root of q and get, I should say this, take the square root of q squared to get q, right? I have no idea why this is fuzzy, but you can still see it. So taking the square root of q squared gives you q, which is 0.5152, which is what we started with. Well, of course it's true, right? Just all we're doing is undoing what we just did. So it puts us right back to 0.5152. So this number here, where the question marks are, is 0.5152. So that means what's p? Well, remember, p is 1 minus q. So we take that and we get the exact same number again. So even though the genotype frequencies changed, the allele frequencies did not. Therefore, this population did not evolve. How can that be? Well, actually, it's not that surprising. What you got here are 3 equations and only 2 unknowns. P and Q are the 2 unknowns. You got 3 equations. So there's an infinite number of solutions, and there's an infinite number of ways that you can combine these 3 frequencies with these 2 allele frequencies. So these don't matter. That's why we define evolution the way we do. These don't matter. What matters is this. This didn't change. Okay, so what happens in the next generation? Well, next generation, if those 5 assumptions still hold, then this number plus plus is going to be p squared, which is this number, which is going to be that number again. So it'll be 0.2350, same thing. What about this? This will be 2pq. So it's going to be 2 times 0.48 times 0.52, which is the same as this number. So that's going to be 0.4996. And this is going to be q squared. So it's going to be 0.2654 again. So what about the next generation? What about 500 generations from now? What about 1,000 generations from now? If those 5 assumptions do hold, then these will not change. They will always be the same forever and ever and ever. That means this. If those 5 assumptions hold, you do not have any evolution at all. Evolution cannot occur.